So today I'm going to tell you something about my postdoctoral research. It's been something that I have been carrying out in Strasbourg over the past 3.5 years. But in order for me to tell you what it actually was, we need to go back in time by about 4.5 billion years. 4.5 billion years ago, there was no life on Earth. But what we know was true back then, as is true nowadays, is that in the core, you had lots of iron. It was metallic iron. And then the further you went outside through the mantle towards the crust, the more oxidized the iron would become. You have iron minerals, iron +2 and iron +3. This creates an imbalance, which you could compare to a giant global Earth-scale iron battery. So remember this fact. Before we move on further, I have a little demo. So in your hands, different forms of iron that I work with and different forms of iron that you can find on our planet. Please return it to me at the end of the day. Take your time. Don't open those vials. I'm not responsible for any casualties. Anyway, what we know was present on Earth before there was life. Lots of rocks, different minerals containing metals such as iron, like the ones that you have in the box. Atmosphere made mostly of CO2, according to most models, carbon monoxide, carbon dioxide, probably also a lot of hydrogen, maybe some other gases in smaller quantities. There was no oxygen, or very little. Then what you have here, this is life, this is biochemistry, this is a map of life, different transformations between life's biomolecules that are organized in those really funky patterns. This is a really beautiful organization. So where I work is somewhere here on this interface. We are trying to explain how those inputs, inorganic, dead, abiotic inputs, from the early Earth, how they got together and self-organized into something that we see today in all the living cells, those patterns of biochemistry. The origin of life is not just a philosophical question, because if you really want to say that we understand life, biological life as it is, if we cannot create it, we don't understand it, to quote the classic Richard Feynman. So we are somewhere here. A lot of people who work in this research focus on making the building blocks, the biomolecules. But I don't really believe this is the only right way to go, because life is a disequilibrated open system. It follows the organization pattern of such phenomena as, for example, a hurricane. If you take the building blocks of a hurricane, air and water, and you put them together, you obtain air and water. You don't obtain a hurricane. If you want to get a hurricane, which is the process, you need to apply an energetic stress, different temperatures and different pressures. And the same is true about life. The building blocks of life include carbon, hydrogen, oxygen, sulfur, nitrogen, phosphorus, different metals, different small molecules. But if you put them together in a vessel and shake them, you obtain a goo and not life. For this reason, we also need to look at different gradients and different stresses, different sources of energy. So, for example, redox gradients, such as the one in the Earth battery, or light, or different sources of geothermal energy. And only then do we get to real processes, which in biology are reflected in metabolic pathways. So life's processes are just as fundamental to the nature of life as life's building blocks. So how is this affected in biochemistry today? So you have this little organism, and it transforms biomolecules following those organizational patterns. And to do that, it uses enzymes, like the one here. These are very complicated molecules. For sure, on Earth, they were not just like, anywhere on the ground. But one curious thing about enzymes is this little green blob is a metal, that's nickel. And the red thingy, tiny red thingy, this is iron. So maybe this is trying to tell us something. Also, pretty often, enzymatic processes, they use energy. This is the biochemical energetic currency. This is ATP. For sure, there was no ATP on the early Earth. So how does this come about from this? Well, that's where I get to our working hypothesis. So what if I told you that metabolic pathways emerge spontaneously, driven by the inputs from the environment, driven by environmental metallic catalysts that were not enzymes, but they had the same metals that nowadays enzymes do? So how did we get to actually make it work? We started reading papers. This is a really good thing to do, read papers. This is a paper from a bioinformatics group in Boston from Dan Gray. And what they did is they looked at the entire KEGG database. This is a database of all the biochemical pathways. And what they found is that the most populated nodes are these two. If you have no idea what I'm talking about, think about it as a subway map. So you have stops which are really hubs for the most traffic. So the hubs that transfer the most traffic are here. This is glyoxylate, and this is pyruvate. These two small molecules. Pyruvic acid, biomolecule found in fermenting apples. Glyoxylic acid, a small molecule found in green apples. Apples have nothing to do with the origin of life, by the way, just illustrative. Anyway, we are chemists. So obviously what we did is notice that these two molecules can react together. Again, if you're not a chemist and you're really put off by this slide, stay with me, I will walk you through it. So we are chemists, so we took a little vial and we put these two small molecules together. And we also added some iron, because why not? So we put these two molecules together, added some iron and water, and started heating. This is the only human intervention, in this process. And then as the time went by, we observed all this started developing, all this complexity, all this pattern that is so characteristic of biochemistry and of biology and of life. Moreover, what we noticed that if you add a nitrogen source, you're also able to obtain 4 Life's amino acids. So also from the same system, in one pot, just one human intervention. So this is starting to look like biochemistry without enzymes. Or is it? Let me show you to what extent this looks really like biochemistry. These two cycles, these are biochemical cycles. The red one is a Krebs cycle. You might remember it from high school, from your high school exams. This probably not so much. This is glyoxylate cycle, but it's quite similar. It has essentially a similar purpose. And the thick, bold, colorful lines show, in both cases, the extent to which our network overlaps with what biology already does. So it's a really, really high level of overlap. In the Krebs cycle, 9 out of 11 intermediates, 7 out of 11 reactions, so the transformations. And in the case of the glyoxylate cycle, 8 out of 9 reactions, 8 out of 9 intermediates, and 5 out of 8 reactions. So it's really, really close. So that's how we do it. It's really easy, to be honest. Wear gloves, that's my hand. We screen a lot of reactions. So basically, this is a heating block. These are reactions. High throughput, a lot, at least 50 per day per person sometimes. Then we analyze. We get rid of the iron because it comes in the way. And then we make those little samples. A syringe injects them into the machine, and then we follow on the computer. This is gas chromatography coupled to mass spectrometry. Sometimes, if we need to introduce a CO2, a carbon dioxide atmosphere, we use those here pressure cookers. These are safe. We only evacuated a building one time over 4 years, so I think it's pretty good. And then you could ask, OK, which places on Earth could you find these processes in? And to be honest, we are not really adamant about one particular setting. There are people who support hydrothermal deep-sea vents. These are sort of underwater volcanoes which eject streams of hot water or warm water, different types of them. They also contain a lot of iron minerals in the structure. You could also think about surface pools that periodically dry. They are heated by geothermal sources, by volcanoes. To be honest, I personally don't care. Wherever it works, it works. It's good if it works in more places than one, that's for sure. What I would like to take away from this talk as a final message is that iron minerals and iron salts, like the ones in the box, enable self-organized chemical networks that really mimic today's enzymatic biological pathways. And finally, I would like to thank all those people who were involved in this project. It's been a lot of work. It's been a fascinating journey. So first of all, Professor Joseph Moran, who's been a fantastic postdoc advisor and who is also a fantastic research fellow advisor at the moment for me. Dr. Srijit Varma, who really worked with me on all the projects connected to this one, and all the other people, we are the prebiotic people, and all the non-prebiotic group members that I don't really have enough space on the slide to thank. Also our collaborators in Germany, in Japan, around the world, and people who give us money for us to be able to do exciting things. And finally, I thank you for your attention. I'll be happy to take questions.